All right, good evening. It's time for one more journal club. This is the last, this is number 8. And for those of you in immunology class, we will now be moving on to the final. And this one is a shorter one. It's also a comprehensive one, but it's really cool. It's actually a machine learning one, and it uses a language model just like what we have in ChatGPT. But instead of looking at language, it looks at the language of genes, language of people versus language of genes. Genes are made of letters, and letters can be analyzed like words. Genes are basically words. And so why not use ChatGPT style reasoning on the words that you have in your immune system, because your particular B cells and T cells have adapted. They've rearranged their DNA, and so they have made B cell receptors and T cell receptors that are unique to you and unique to your history. If you have a disease, that should be reflected in the way your immune system has adapted to it. So we can read that out, and we can try to say, can we see who has what disease, not by looking for the disease, but for looking for the disease's impact on the immune system genes. And it's pretty cool because it involves a lot of genes and involves both T cells and B cells, so it brings a lot of things together. The idea is if you sequence enough TCRs, you will see patterns that will indicate, if you see a lot of TCRs that you know are reactive against COVID, you will know that that person had COVID. In fact, you can look for TCRs that are reactive to parts of COVID that were not in the vaccine. And you can possibly figure out there's a few jumps in logic you have to make, so you're never 100% sure. But you can find patterns that will say, you have had COVID if you have these TCRs. If you have expanded this T cell population that reacts with your MHCs in this way, we can say that we think that you are reacting to this COVID peptide. And so there's a lot to untangle, and it's not as simple as just let's look at the antibody sequence and we know it binds one antibody, binds one antigen. You have a lot more going on with TCRs, but it is the same idea that there is a structural rationale that your TCRs have molded themselves to the infection by responding to it and by expanding the responsive TCRs. You can test for that. And there was actually something way back in 2021, a Seattle company called Adaptive Biotechnologies made a proposal to examine T cells to see if they had particular kinds of receptors that would have sequences that would correspond to viral interacting T cells. So it developed this. Actually, it was testing before and after, and it could find T cell responses that correlated with COVID 97% of the time and correlated with not having COVID 100% of the time. Very cool. And they even let it be put out there that you could do this. You could have your T cells tested. And according to their own paper, now this is their scientists, it was a preprint that wasn't reviewed. I haven't checked to see if this got through to a journal. So it might be a little biased, but this claims to have data that the T cell testing is not only as good as antibody testing, but actually better in determining if you have been infected with COVID. Because COVID can do asymptomatic infections, it's possible this is the only way you could catch it. Maybe you can find antibodies that responded to it. Maybe you can find T cells. Maybe if you never had symptoms and you never tested yourself at the right time, how would you know otherwise? And so they actually offered this. I thought about getting it, but I didn't feel like paying $150. And of course, once COVID became less of a problem, I'm not sure if the company is doing that well anymore. I haven't caught up on them. I haven't felt the need to get my T cells tested, but I always thought this was a cool idea. I almost went for it. But it's the idea of sequencing TCRs, and you can also sequence BCRs. Same idea, clonal expansion, right? Clonal selection undergoes in both of those. You have a bunch of T cells that have the same sequence. You have that sequence for a reason. If you can back infer that because you have this T cell, you must have this disease, because those T cells only come from that disease. If you can do that enough time, you can do sequencing to diagnose. Very cool. Just remember, and this is the immunology part of it, that you have like VD and J chains for your BCR heavy chain is what they looked at. Technically, they looked at the TCR beta chain. And that's where they looked at and saw which V genes you had, which Ds, which J, which other mutations you have. And they would be able to correlate particular VD-J combinations with the presence or absence of disease. So that was a yes, no, COVID or not question. This is taking the same idea and is saying, can we sequence them and not just look for one disease, but look for pretty much all the major immune diseases that we have a lot of information on, with COVID-19, HIV, influenza, lupus, and type 1 diabetes. A lot of big diseases, and they had a pretty big patient count. You got to realize this is such a big study that I still have questions whether the patient count is really adequate to so many different diseases. But at least they do have patient counts in the 100s rather than patient counts in the 10s like you would used to have. And the main thing is they're using machine learning. They're asking ChatGPT, are there cases where you can see TCR sequences that go with this disease? Then we'll give you new data, new TCR sequences, and we'll ask you, do you think the people with these TCR sequences have this disease or not? And what they said is they want to predict, and then they have cases where they know that the person has the disease. They want to see how the prediction lines up with what happened. So there's a couple of grids about this. All of these grids have predictions on the x-axis and patient of origin, which means the known disease, the clinical data on the y-axis. So going across, you have the prediction. Going up, you have the actual infection. They're in the same order. So if the prediction matches the infection or the disease, I shouldn't say infection, these are not all infections. If the syndrome matches the prediction, then you will have the place where they line up is on the diagonal. And they will make it darker if a higher percentage of the results end up in that square. This is the overall, this is the diagonal means correctly predicted. And you can see you have a really good number of blue squares on the diagonal. In fact, in all cases, the biggest number in each row and column is going to be on the diagonal, which means it was the computer correctly said this person has that disease or this person is healthy based on just their TCR and BCR data put together. A ton of data, you can't imagine doing it by eye or even by statistical methods, but something like a large language model actually really helps with this. It's one of the good uses of AI. There are some. So you look up and down, you see the number of cases. Of course, they had different numbers. They had a lot more healthy people than they had anything else. That gets the dark blue 160. But I want you to look at the rows. Each row is what the disease actually was. If it's on the diagonal, it means that the disease was correctly predicted. If it is a large number not on the diagonal, that means for that row, the computer predicted that they would have a different disease or health status than they actually had. It's where the computer got it wrong. They also make these slightly darker when it comes to the shade of blue. So look for the biggest number. So look at just look at the diagonal is correctly predicted. Look for squares that are off the diagonal that are shaded blue / bigger numbers. And I want you to, if you look at the row, look at the healthy row and look across and look for the two biggest numbers that are not the 160 number. You can see right here, patient of origin, healthy, there were 15 patients of origin that were actually healthy, but the computer said it thinks they had HIV. Move over to the second biggest number for healthy. The other mistaken disease is 9 patients. The computer said they had lupus when they did not. Likewise, you can actually take lupus and go over to healthy, and you can see only 4 patients were predicted to be healthy when they actually had lupus. But this is actually a relatively big number. The main thing I want you to see is where was the computer wrong when people were healthy and it said that they had something. Is for HIV and lupus. 2 very tricky diseases that involve hiding out in your own immune cells for HIV, and attacking your own antibodies for lupus. Interesting. The other diseases were much better predicted. The other thing I want you to do is look at the purple square. This is the disease when you're looking at the column for healthy people. This is the people that the computer said was healthy but was not. You see the healthy column, but it was in the influenza row. Now notice that I circled influenza for this and not lupus, even though they had the exact same number. The influenza samples are smaller. You see there are only 31 correctly predicted people, and if you add up the numbers, you see that you have less than 40, whereas lupus has more than 40 people in it. So getting 4 incorrect diagnoses of where you say an influenza infected person is actually healthy, that's actually worse for influenza than getting 4 wrong out of a larger group. Getting 4 wrong out of 40 is worse than getting 4 wrong out of 60. See how that works? You can also tell by the shade of blue, although at this level it's better to go by the numbers. I want you to be able to look at this and say, okay, the computer predicted this, they actually were that. Okay, how bad is that? And to see overall, the computer's pretty good. You know, most of the people are predicted correctly. I don't even know what I said there. So the one thing they were wondering is, for a couple of these diseases, now they only had data for adults for influenza, HIV, and COVID. They only had adult data. But they did have children data for healthy, for lupus, and for type 1 diabetes. And so I'm going to block those off. And this is actually the same graph, but it's just separated out into 2 rows for each of the times they had child data versus adult data. So you can see the healthy, they had about 40 children, and 32 of them were correctly predicted. The diagonal is still there. It's just sort of stacked up on top of each other for the child versus adult data. You can see the 32 is still for under 18. It predicted that they were healthy, but look at this. It predicted that 9 of the healthy children actually had lupus. It predicted that they had lupus when they didn't. And on the other hand, for the 18 plus category, it predicted that they actually had trouble detecting the disease because it only detected 12 of those when there were a lot more. There's 7 on the one side, there's 4 on the other side. So only predicted for adults with lupus, it only predicted that they had lupus right 12 out of 23 times. The other times it got it wrong. And so that means it's a light square for that. You see under 18, it got it right 40 out of 41 times. So you see there's a real divide over whether the lupus people had the computer could detect lupus in adults, whereas it could detect it in children just fine. So which disease were healthy children inaccurately diagnosed with? I mentioned this before, but this is where you have that square up there, the purple square. If you look at the healthy under 18 row, then you will see that the children were diagnosed inaccurately with lupus, even though they were healthy. It looks like lupus is giving the computer problems. It's being wrong in 2 different ways here, in missing adults with it, and misdiagnosing adults, and in misdiagnosing healthy children as having had it. It's like missing adult cases and giving it to children cases that don't have it. Even this is actually, it's pretty good. You'd expect one of these to give it a problem. And again, lupus, if you think about the way lupus is, the kind of disease it is, you're inclined to give the computer a break on this. They even had a whole slide about this because lupus was the thing that gave them the most trouble. This is where they actually went, and rather than saying they had a score that gave them lupus, yes, no, do they have lupus or not, they went and they looked at the actual scores that the computer added up to say as a measure of how certain the computer was. A high score would mean the computer was very certain. A low score would mean it was less certain. And so what they did, all these patients actually had lupus, but the green ones were actually misdiagnosed as healthy. This would be the red square on this, lupus 18 plus. The computer says it would be the things outside the red square, where the computer says it doesn't have lupus, but they actually do. The green ones were misclassified as healthy, and you see that there's a number of them with a low score, but there's a considerable number of them that actually had a reasonably high score that was, maybe what they should do is they should just let the score be a little bit more clear. But also the adult data for lupus, maybe the adult scores are low. Maybe the green lines, you could draw a line that would include more of the green lines. But maybe also the lupus responders just don't leave as much of an imprint on their TCR, BCR data. And by the way, there's another wrinkle that some of the adult lupus patients, they only had BCR data for some reason or another. So this is basically, you see the thing is there's healthy people with a somewhat high score, but more than that, there's a lot of lupus-containing people with low scores. In fact, there's a lot of overlap between healthy and lupus. So, and the other thing to remember, lupus is SLE. The L in SLE means lupus. The technical name for it is SLE, and you see that here. So if you look up and down at this, and I want you to look at this, they think they have an explanation for why lupus is so hard for them to catch. The misclassified patients has lower systemic lupus, I can't even say the E, scores. Possibly because their treatment actually changed their B cell repertoire so that it became more normal. It's possible that the treatment resulted in B cells that were more normal, and therefore the computer, which was only looking at the B cell receptors, had trouble finding them because their treatment had reversed it. And this also is something that would happen more for adults than for children. And that's what they saw. They saw this direction of misclassification in adults rather than children. So they also looked at, and they also found that even though they didn't have much TCR data, there was actually 85.3% overall accuracy when you use BCR and TCR together. More data means better accuracy. We want to look at each of these diseases because what they have is they have the graph I previously shown you on the left, then the graph in the middle is BCRs, and the graph on the right is TCR only. So the graph on the left is using both BCR and TCR sequences. The one in the middle, only BCR. The one on the right, only TCRs. And so the question is, if you look at this, there's lupus with both. But if you look at lupus with BCRs, it's darker, it's a higher number. 65 were correctly classified as being on the diagonal. They had lupus, the computer said they had lupus. And if you look at the TCR only data, it ends up being 46. So you can see that the BCR only for this actually ended up being better for lupus. By the way, the number I quoted on the last slide, I believe that's overall accuracy. Overall accuracy is about 80%, but then we can break it down by disease and we can see these things in another way. So I want you to look at type 1 diabetes. What is the best? And these numbers are all the same, so you can compare them directly. The highest number is the best result, the highest number on the diagonal. Best result, lowest number, worst result. So just take a second and look at it. If you see the highest number for both BCR and TCR data, good. That means that actually worked best. This is the typical result. If you look back and forth, the highest numbers tend to be on both data types, or they tend to be the same. Like for influenza, they tend to be the same. But if you look at the BCR and TCR numbers, they're both lower in the middle for BCRs and on the right for TCRs. That means the best performance, the most accurate diagonals, the highest number on the diagonal is on the left here. And you can do this for any disease. The important thing to remember is, like for COVID, 49 with both, 50 with BCRs, and 45 with TCR. So TCR only is definitely worse. BCR, it's 1 point above. That's pretty much the same. Oh, and here's one to look at. This is the type 1 diabetes. That's actually, actually is a healthy person that is misdiagnosed as having type 1 diabetes. If you had the BCR only model, you get a huge number relative to the number of people. You get like 33 false positives. And also, if you look at healthy background, on the other hand, for TCR only, you get a huge number of false positives for HIV. If you look over at these numbers on both, the 33 for BCR only for type 1 diabetes, the purple square, goes down to 6. The 31, the green square for TCR only HIV being misdiagnosed, that goes down to 15. Still, it's tricky to catch HIV, but it works. I'm pretty happy with this, with the versatility that's showing. So now we can divide it by age, sex, and ancestry only on the left. Now this is the question of how much is sequence doing versus other things, everything else that we can measure easily, which is age, sex, and ancestry. And so what they did is they split this out. The age, sex, and ancestry is only on the left, and you see there's a lot of blue squares. There's a lot of wrong answers if you're only considering age, sex, and ancestry data. If you're only considering sequence, it's actually pretty good. You have blue lines on the diagonal, blue boxes on the diagonal. And if you consider, well, sequence only is in the middle with everything. I should say is sequence plus age, sex, and ancestry in the middle. So it's sort of both, and it's actually sequence only on the right. You see the sequence only on the right, it's about the same, maybe a little worse. If you look at the numbers are a little bit lower on the diagonal than they are. This shows that sequence is the major effect. Age, sex, and ancestry are secondary effects. And so you can ask, the sequence only is actually a primary effect. If you're making a diagnosis or even a statement of causation that is based on age, sex, or ancestry, you are working on a secondary effect than what's really the primary driver, which in their cases, the primary way of diagnosing these is sequence only. It's better than all of the other easier to get things. This says to me that race does not matter that much. So that's an interesting conclusion from this. Do not conflate race with immune genes. Your immune genes are influenced by the environment. There's some aspect of them, and in the paper they even show you there are some aspects of the immune genes that you get that are associated with what we classify as race, but not many, and they aren't as important as other things. Race genetically is a secondary category that does not affect as many things as we think it will affect. That's actually a very interesting result for this, and it's why when we talk about immunity, we often don't talk about race, because it's a secondary effect. It might have some effects in some things, but it's not as big of an effect, and we have a human problem that we amplify it when we shouldn't. It really is, at least for this immunity, it really is skin deep. Again, that's to say, maybe there's some places where it matters, but the more different genes you have going on, the less race matters for those things. And honestly, most of the important things in life are polygenic. They have a lot of genes going on. And so I think that's an important thing about how race is less important than we might think it is. So those are all really great things to reason through this. I want you to think about this. I'm going to give this to you and have you interpret it, and then we're going to see it again on the final. This is a great way to review and think about all these things and all these different diseases at once. Each of them is different, but they were all predicted mostly correctly, like 80% above correctly by the computer. So that is our journal club.